Hello everyone. Adab, Sat Shri Akal, Namaskar. Myself Priyanka Rao. Welcome to Raw Biology classes. Here we all know that we starts the chapter Cell, the functional unit of life from class 11th. In this chapter we all learns about the endomembrane system. So guys, today we continue this topic endomembrane system. Endomembrane system, as we all know, that involves the four organelles. These organelles are endoplasmic reticulum, Golgi apparatus, and lysosomes. So today we discuss the lysosomes topic. Here the lysosomes. Let's start the guys lysosomes. So first we understand the meaning of this word lysosomes. This lysosome word is made up of two words, lysis, and the second one is some. Here the lysis word meaning is digestive and some means that body. So by combining these two words we call the lysosome word means that digestive body. So the word digestive body indicates that there is something which contains lysosome which help in digestion of food or any other particles which are present in your cell. So today's the digestive body contains some enzymes. These enzymes we call that hydrolytic enzymes. Hydrolytic enzymes. Because they mainly functions in acidic medium. Okay, this all about the outline of lysosomes organelle. So guys, first we discuss the definition of this organelle. As we all know that the organelles which are involved in endomembrane system all are single membrane and the lysosome organelle are made from Golgi apparatus in the form of vesicles. So it formed in the form of a small vesicles and it is a single membrane. And the third point, as we all know that it contains hydrolytic enzymes. So by combining these 3 points, we define the term lysosomes. Okay, so the lysosomes defined as it is a small vesicle which is single membrane containing hydrolytic enzymes which help in digestion of food or any other particles present in your cell. So simply we define the lysosome by combining these 3 terms. Okay, this is the definition of lysosomes, and secondly, we study the discovery of lysosomes. The discovery of lysosomes is performed by a Belgian scientist by accidentally when they perform a fractionalization technique. So fractionalization technique guys is not your syllabus, but I can explain that a little bit. Here the fractionalization technique is that by which we separate the cell's components. Okay, cellular components. For example, mitochondria, ribosomes, lysosomes, chloroplasts on the basis of their size. So, lysosomes is also separated or discovered by the fractionalization technique. So, lysosome is discovered by a scientist which we call that Belgian scientist is Christian D. Dew in 1955. This was the scientist which discovered the term lysosomes, but there is another scientist which gave the term of lysosomes in the next year is Novikov in 1956. He is the one which gave the term of lysosomes. Term is given by him. Okay, these are the two which involves in its discovery. The most important point is here that the D due. In most of your exams, the options are come in the form of short form D due, the discovery of lysosomes. So, we learns the definition and the meaning of the lysosome, and the second is we learn about discovery. Third, we learn the location about location. Lysosomes help in digestion, so they basically present in animals, plants, fungi. But when they are present, there is an exception is also occur. They are present in animals in the form of lysosomes, animal cells, but there is an exception here is that it absent in RBC. Okay, and in plants and in fungi, their function is occupied by vacuoles. Okay, mainly in animal cells, it present in abundant form in phagocytic cells, the cells which are phagocytic in nature. These cells are, for example, Kupffer cells, which are present in liver, leukocytes. Okay, mainly in these cells, these are present in abundant form. This all about the location where the lysosomes are present. And the fourth point is that the another name of lysosome is suicidal bag. Suicidal bag. Okay. Why we call that suicidal bag? Because there is a reason behind this is the lysosome contain the hydrolytic enzymes. As we all know that these hydrolytic enzymes help in digestion. Okay. So which we you call that suicidal bag, or the material or any other substances which are not useful for the cell are digested by lysosomes or removed by lysosomes. So we call that suicidal bag. And the most important point is that we continuously discuss the hydrolytic enzymes. So what are these hydrolytic enzymes? So guys, these hydrolytic enzymes involves acid phosphatase, sulfatase, nucleases, proteases, peptidase. These are the enzymes which are acidic in nature, and these involves in hydrolytic enzymes, and they are functions in acidic medium, mainly acidic medium, and the pH of this acidic medium is 4 to 5. Okay. For example, the nuclease enzyme digests the nucleic acid, protease enzyme digests the proteins, peptidase enzymes digest amino acids. Like that, there is a carbohydrases which digest carbohydrates, or many more. So these are the enzymes which digest your food components, these which are present in your food material or substances. So there is a question here is arises here is that what by how the acidic medium of lysosomes is created. The acidic medium of lysosome is created by the pumping of protons, pumping of hydrogen ions, or we call that protons, into the lysosomes. When the hydrogen ions, or we call it protons, are pumped into the lysosome from the cell, there is an acidic medium is created. This acidic medium is help in functioning of hydrolytic enzymes, and the hydrolytic enzymes are help in digestion of food which are present in your body or cells. So, the all about the lysosomes. We discuss the meaning of lysosomes, definition, location, and the hydrolytic enzymes basically, and the discovery. So let's we discuss about the type of lysosomes. Basically, the lysosomes occur in our cell in different forms or exist in different form. So we call that lysosomes are showing polymorphism behavior. Polymorphism behavior, which means that when anything exists in our cell or anywhere in our environment in different forms, or in their lifetime they exist in different stages. So we call that they are polymorphism in nature, polymorphic in nature. Okay, so the lysosome is of four type: primary, secondary, third is residual body, fourth is auto lysosome. Basically we call that auto lysosome in simple words. Okay, these are the four type of lysosomes or four forms of lysosomes which occur in our body during their lifetime. Okay, so let's we start discussing the type or form of lysosomes which occur in our cells. So for example, this is the cell. Before we discuss the lysosomes forms, we discuss something in the lysosomes basically formate from endoplasmic reticulum and the Golgi apparatus, both combined activities. The lysosomes are formed. Okay, I was discussed in my previous videos endoplasmic reticulum and the Golgi apparatus. Okay, the lysosomes contains hydrolytic enzymes. These hydrolytic enzymes precursors are formed firstly in endoplasmic reticulum. Okay, for example, this is an endoplasmic reticulum. Here there is a rough endoplasmic reticulum and smooth endoplasmic reticulum. This is rough endoplasmic reticulum and this is smooth endoplasmic reticulum. Okay, and here is, we call that Golgi apparatus. Okay, when the precursors of hydrolytic enzyme first is formed in rough endoplasmic reticulum, they are transferred into the forming phase. This is the forming phase. Okay, forming phase of Golgi apparatus. In the forming phase, the precursors of hydrolytic enzyme are transferred through small vesicles. This all I was discussed in my previous videos in a detailed form. I was discussed here in a outlined only. Okay, so these vesicles are formed, contains is the precursors of hydrolytic enzymes. They are transferred to Golgi by fusion or through SER. When these vesicles fuse into the Golgi apparatus, they are transferred to Golgi apparatus. Here the packaging of hydrolytic enzymes are occur. So when the packaging is complete from their maturing phase, maturing phase. This is transferred in the cytoplasm in the form of vesicles. These vesicles we call that lysosomes. Okay. So when the newly pinched of vesicles are formed, we call that this is a primary lysosomes. This is primary lysosomes. These primary lysosomes are small in size and they are newly pinched of lysosomes. They contain hydrolytic enzymes. Okay. So, simply the primary lysosomes are the newly pinched of vesicles from the Golgi apparatus in the form of Golgi vesicles. When these Golgi vesicles fuse with endosomes, they form primary lysosomes. Okay. Suppose this is endoplasmic reticulum. Here, the small vesicle is formed in the form, they contain hydrolytic enzymes in a small vesicles की form में primary lysosomes form हुआ. When these primary lysosomes further fuse with phagosomes. What is this phagosome? These phagosomes contain solid food particles or solid substances. When they fuse with these primary lysosomes, they form secondary lysosomes, or we call that heterophagosomes. In these secondary lysosomes or heterophagosomes, digestion is occur. When the digestion is occur, the digested food particles are fused into the cytoplasm and they left or contain undigested or indigestible substances. When this contain indigestible substances, we call that residual body. Okay, this residual body further are transferred or thrown out from the cell by exocytosis process. Okay, this is exocytosis process. Here, these residual body are thrown out from the cells because they contain indigestible substances. This process is occurred through exocytosis process, or we call that this process is exocytosis. Here, this exocytosis is performed by when this residual body is fused with plasma membrane. They are thrown from the cell. The process we call that exocytosis. Okay, this is all about lysosomes, but the fourth one is autophagosomes. Here, the autophagosomes like autophagosomes or autolysosomes is formed when the primary lysosomes are fused with either their surrounding organelles. They form autolysosomes. So guys, this is all about the four type of lysosomes or the lysosomes occur in our cell in their lifetime. And the last topic is function. I was discussed about lysosomes. They contain hydrolytic enzymes. So here the most important function is that they help in digestion of your food, or simply we say that it help in digestion. And second one is that they help in removing of undigested food or any other toxic substances which are not useful for our body. They also help in aging, removing of they help in aging. Means that removal of cells which are not helpful for you, or which performs or the cells by which we causes aging. Okay, so they perform apoptosis. Apoptosis means that programmed cell death. So this process is performed by lysosomes also. So there is many functions which is performed by lysosomes. So I hope guys this topic is clear today, and I will start it in my next video vesicles, which is the last topic of endomembrane system. If you have any problems or queries, please comment and in my comment box. So thank you so much for listening me carefully. Thank you so much.